Welcome to Lecture 8 of Section 1. We are going to pick up where we left off with regards to neurotransmission between neurons. We talked a lot about the synapse last time and to talk about different characteristics of neurotransmitters. This lecture we're going to talk about different types of neurotransmitters and the background behind them and then what they bind to. So we're going to spend a lot of time talking about the receptor, which is important, and we're, like I mentioned, a lot of time, that's the focus of this lecture, as well as then how the neurotransmitters actually influence the brain and the different types. The small molecule neurotransmitters were the first to be identified, and one of the things that's interesting about them is that they are readily synthesized from our dietary nutrients. What do you think is important, or why do you think that's important for you to understand that with regards to our diets? If our diet is the source of the backbone of these neurotransmitters, it is important that we have a varied diet where we get all these different types of things in. So for example, we're going to talk about acetylcholine. So acetylcholine is literally the joining of choline and acetate. Choline comes from foods like avocados, olives, that come from nuts, salmon, those types of things. And acetate is created when you break down acidic foods like lemon juice or vinegar and those types of things. So these two things you have to have in your diet. And what happens is there are enzymes that come in and make changes, transformational changes to these items so that they will end up binding together and creating acetylcholine. Now it's important to understand, I don't care that you memorize the enzymes in this class, but I'm providing you that information in case you're interested in them. Now the other thing about acetylcholine, and is true about all neurotransmitters, what do we know about the importance of breakdown of a neurotransmitter with regards to the synapse? If you're thinking, well, it's more than breakdown, it is also removal is really the most important thing. That is the accurate answer. So at the end of the neurotransmission, after the synapse, the message has been sent from the post to the pre, the system has to be shut down. Otherwise, that neurotransmitter will continue to bind and release from the receptors and will continue the neurotransmission, right? And neurons don't want to be constantly stimulated. So for acetylcholine, that's actually broken down by acetylcholine esterase. It is a enzyme that degrades the acetylcholine into choline and acetate, breaks it apart so that it can no longer bind to the receptors. Here's a quick picture just to talk you through those pieces. Here you have the acetate on acetyl-CoA. The enzyme comes in and cleaves off the acetate, puts it onto the choline. Now you have acetylcholine. It gets released into the synapse. It comes in, it binds to its receptor, comes off, and then acetylcholine esterase, the enzyme, will go in and cleave off the acetate so that this structure can no longer, choline by itself cannot bind to the receptor. And then it is then absorbed back into the presynaptic as acetate and choline. Kind of cool. It's a nice little system that's designed. Things to think about when you are processing how this information is actually affecting the way the body functions. What do you think are rate limiting factors with regards to neurotransmitter being synthesized? Well, there are two things. There is a limited supply of the enzyme that helps create or synthesize the neurotransmitter. There's also a limited supply of the basic molecules that you need to create the neurotransmitter, depending on your diet. If your diet is low in acetate or low in choline, no matter how much enzyme is present, if it doesn't have its basic needs or the basic compounds it needs to combine, it's going to create a problem. And I just want you to see that many of the neurotransmitters are actually linked in the way that they are created. So tyrosine, which is a chemical amino acid from your food, gets transferred into L-DOPA, which then turns into dopamine, which then can, if there's another enzyme present, becomes norepinephrine, and then and then epinephrine. So as you can see, they're all kind of connected. I'm going to have you take a break, and I want you to watch these two clips. If you've seen I think it was the movie Awakening, it covers this historical event. But basically, it talks about the role that a flu created in people who may not have died from it, but were severely damaged from it, and it has to do with where the virus actually attacked in the brain. And it's interesting to see just how important neurotransmitters are. So take a break, go watch those two clips. They're very fascinating. Welcome back. Hope you enjoyed the videos. And now we're going to talk about some specific amino acid transmitters. And these are two that have very specific and broad effects on the brain. And these are the only two that I will ever ask you, what is their effect on the brain or effect on neurotransmission? So glutamate is the main excitatory neurotransmitter. That means it is the almost 99.9% of the time glutamate is released, it will cause the next neuron to be excited. I mean, sodium will go in, calcium will go in, those types of things. That's going to happen, and it's going to increase neurotransmission. GABA is the main inhibitory neurotransmitter. So when GABA is being released and activating its receptor, it is going to cause choline to go in, which is going to hyperpolarize the cell, which means it is less likely to have have an action potential. These are the only two, I'm going to say it again, these are the only two neurotransmitters that I will ever ask you, what is their main effect? So glutamate's excitatory, GABA is inhibitory. And they're actually created from one another. So glutamate is the precursor to GABA. So very structurally similar, and given that they are both the primary neurotransmitters that the brain actually uses, it's nice that they have a similar background. The other major inhibitory that I like to give people is glycine, but that is really only present in the brainstem and the spinal cord. You won't see that too often, but it does have a very inhibitory effect, and is actually pretty active during sleep with removing motor control. Moving into peptide neurotransmitters, these are things that are slightly larger than the small molecule peptides that we just got done talking about. They are synthesized from your DNA via messenger RNA, and this process is slower. So you have to think about neuromodulators, things like oxytocin and substance P, which has to do with pain, those types of things. They often don't bind to ion channels, but they have an impact via an indirect influence, which may help neurotransmitters bind more quickly, may modify the way in which different neurotransmitters are successful at creating their environment or their effect. They often are hormones and things when we get to eating, and a lot of the things that you think about psychology with regards to pleasure and pain and those types of things, neuromodulators play a more important role. Here's an example of just a list of different neuromodulators. Not going to spend a ton of time, don't need to memorize all of them, but you may recognize some of them. So vasopressin and oxytocin, those are things that you may have heard in other classes. Glucocorticoids, we're going to talk about when we talk about stress. And then insulin and some of these other somatostatins play a role in eating behavior, and we'll talk about that. As I mentioned, you can be using either the 4th or 5th edition of the textbook. This is new in the 5th edition, so all of you who have the 4th edition, please pay attention. There is a new class of neurotransmitters, and those are lipid transmitters. Those include endocannabinoids. Does anyone have an idea of what that is? Endocannabinoid, what do you see in that word? Maybe you see the word cannabis, and that is an accurate statement. Anandamide and the 2-AG, those are our endogenous endocannabinoids. Marijuana or cannabis activates this system, and the receptors are called cannabinoid receptors. They're found in all different types of synapses, but definitely are found at glutamate and GABA synapses. And what's interesting is we don't fully understand how they function, but we're starting to get evidence that they're a retrograde neurotransmitter. When you're thinking about the synapse, there were two ways in which the presynaptic cell was shut off. One was by an autoreceptor, which is the neurotransmitter binding to a receptor that's on the presynaptic neuron to remove or to stop or send a message that, hey, we've got enough information, enough neurotransmitters in the synapse. The other way was a neurotransmitter being released from the postsynaptic to the presynaptic. That's called a retrograde neurotransmitter, and basically shuts that presynaptic off. There are also transmitter gases. Don't spend a ton of time on them because they don't have a major influence on behavior like all the other systems, and we don't have enough time to study all of them. But we do know that there are times when we really want nitric oxide to be present in our vascular system because it plays a role in dilating and controlling blood vessel walls. So an example is the penis. To have an erection, you have to have a male has to have nitric oxide, which causes the arteries to dilate, but not the veins, which allows the erection to occur. And this is actually how Viagra works, by increasing nitric oxide. Just a little piece of information. All right, moving into the receptors, it is important to understand that there are two primary receptors that you should be aware of. One is an ionotropic receptor, and this is one where the neurotransmitter binds at the receptor site, and immediately a conformational change happens in that receptor, which creates an opening in the cell membrane and allows for ions to cross. So it could be sodium and potassium. It could also, which trigger excitatory, or it could be chloride ions, which creates an inhibitory. So let's look at a picture. Neurotransmitter comes in, binds the receptor, the receptor changes shape, and that creates an opening and allows the ions to pass through. The other major receptor, the second type, is called a metabotropic receptor, and this one requires additional communication to occur for the cell membrane to become permeable to ions. And this requires, so it's an indirect messenger because it's using a secondary messenger, which is a G protein. So G proteins consist of 3 subunits. There's an alpha, beta, and gamma. And in this unit, what happens is when the neurotransmitter binds, the alpha subunit dissociates and it goes off and does something else. So it may go interact with a receptor or an ion channel near that receptor to cause a change in that ion receptor, in that ion pore. Could go off and have a secondary effect on some other messengery system. It might go affect the nucleus. So let's look at some pictures of this. Here you have the metabotropic receptor. The neurotransmitter comes in and binds. It activates, changes conformation. The alpha subunit branches off, and it actually interacts with this ion channel, and it causes a change. And lo and behold, the ions are able to pass through. The second way is how neurotransmitter binds. It has the change that occurs. The alpha subunit again dissociates and goes off and has an impact. So it can either go off and interact with a secondary messenger, which then can go affect the DNA, can create the formation of a new ion channel, or it could interact with an ion channel to open. So again, lots of kind of flexibility. Now, in my mind, it's really helpful to watch videos and see these receptors acting in a more active way as opposed to just looking at pictures. So I suggest you go watch these 2 videos. They are very, very helpful. They also are on the list of helpful videos in the Canvas page.
G-protein-coupled neurotransmitter receptors do not have ion channels as part of their structure. Instead, they function through intermediary molecules called G-proteins. These receptors also have 2 domains. As before, an extracellular domain binds neurotransmitters. However, here, an intracellular domain binds to G-proteins. Neurotransmitter binding activates the receptor, which in turn activates the G-proteins. G-proteins then dissociate from the receptor. At this point, G-proteins can either interact directly with ion channels or bind to other proteins, such as enzymes, that go on to open with ion channels. Most of the biogenic amines and peptide neurotransmitters signal through G-protein coupled receptors.
Important things to note. A neurotransmitter is not associated with a single type of receptor. So a neurotransmitter can interact with both an ionotropic or a metabotropic receptor, so it doesn't matter. And the effects can be different. So if it binds to an inotropic receptor, it might have an excitatory effect, where when it binds to the metabotropic receptor, it has an inhibitory effect. So here's an example of one of those. So acetylcholine, we've talked about it on and off. Its role in muscle contraction is to stimulate and cause contraction. That is done through an ionotropic receptor. However, on the heart, the receptors are metabotropic, and that causes the decrease in heart rate. So it's inhibitory. When you're talking about how does a neurotransmitter impact behavior, you have to be looking at how much is released, you have to be looking at what receptor it's binding to, where the receptor is located, and all these pieces. So this is why it's really difficult for me when I say, serotonin, how does it affect the brain and behavior? You're never ever going to be able to give me a single answer, a single sentence answer, because it is way too complicated. Other pieces to understand and why this is so complex and also allows for such flexibility in our behaviors. One is that a single neuron may use one transmitter at one synapse, but can use a different transmitter at another synapse. They're very talented. They also have same coexisting neurotransmitters at a synapse. this is why we cannot, again, reiterate, you cannot make, and when we do this as a society and simplify it, and the perfect example of this is serotonin creating depression. We have created that link in the eyes of lay people, and it is not inaccurate. It's not that easy. It's not that simple. Because there is no connection, there's no cause and effect relationship that we can identify with any neurotransmitter toward the behavior. It is way too simplified to do that. That being said, remember there are two neurotransmitters where we do know the effect of that neurotransmitter on the next neuron. And those two are glutamate, will often be excitatory, and GABA is going to be inhibitory. Those are the only two. Everything else is a free-for-all. The somatic nervous system, we've talked about it, if you guys remember, but this is the voluntary nervous system. This is one we have control over. So this is going to be the muscle contraction component of our nervous system, and acetylcholine plays a major role. We've talked about that a couple different times. In the acetylcholine binding to nicotinic receptors, and the reason why they're called nicotinic receptors, nicotinic acetylcholinergic receptors, is because nicotine, which is in tobacco, also binds to this receptor. But what this does is it allows the depolarization of muscle fiber and allows contraction to occur. In the autonomic nervous system, what's interesting about this is that when we're talking about the autonomic, which is the automatic nervous system, there are two major branches. Do you remember what they are? There's the sympathetic and the parasympathetic, right? So when it's leaving the central nervous system, the cholinergic neurons are feeding into the peripheral nervous system. That's what this PNS is here. So for the sympathetic nervous system, the sympathetic branch of the peripheral nervous system, the central nervous system uses acetylcholine to activate this pathway. However, it transitions over to norepinephrine at the site of activation. And this happens at the sympathetic ganglia, where the neurotransmitter to be used at the site of the organ to tell it what to do transitions. So norepinephrine is actually responsible for causing increases in heart rate, increases in breathing rate, and those types of things. Increases in blood flow to the muscles, that stuff. All that is driven by norepinephrine. The central nervous system also uses acetylcholine to activate the parasympathetic nervous system. But what's interesting is the parasympathetic does not transition its neurotransmitter when the communication is occurring. It remains acetylcholine. The neurons do not switch the chemical messenger. So at the site of the organ, it's flooding it with acetylcholine. And that is the only way the organs know how to behave. Are they being flooded with norepinephrine or acetylcholine? And that's how they change their, the activation, right? So the heart is a perfect example. We all have experienced a racing heart. When we're exercising, it makes sense. When we're sitting in our car, it doesn't, right? So you can picture that happening. So if your heart's racing, that means norepinephrine is being flooded over the heart, and acetylcholine is not. And then as you are able to deep breathe or calm yourself down, your acetylcholine is then flooding the heart at a greater rate, and it's causing a slowing down of the heart. So here's just a quick pictorial example so that people sometimes would like pictures, right? So the central nervous system is always using acetylcholine to communicate its message from the brain to the spinal cord. Once it leaves in the sympathetic nervous system out of the spinal cord, there's the sympathetic ganglion, which is really close to the spinal cord, where it transitions over to norepinephrine. In the parasympathetic, it does not do that. It has the neurons leave off the major vagus nerve and don't have their booster until closer to the organ, which is why it's here, and it does not transition or does not change the type of neurotransmitter. So a unique way in which our brain can use neurotransmitters to drive behavior, and then how our body adapts to change the message at our organ level. These next few slides, we're going to run through the different types of activating systems and the roles or the types of behaviors that they're often linked to. But remember, it's not a one-for-one ratio of how the systems have an impact. So there are 4 major activating systems that we're going to talk through very quickly. These are neuropathways that are coordinated activities in our brain, and they have to do with a single neurotransmitter. One of the things you're going to notice is that many of them follow a similar pathway. However, their origins are different. So the 4 systems are cholinergic, dopaminergic, noradrenergic, and serotonergic. And we're going to talk about all this. So the cholinergic system is responsible for keeping us awake, having our ability to focus, and our attention and memory creation. Very important. We'll talk about this in section 3. Loss of these cholinergic neurons are associated with diseases. The one you are probably most familiar with is Alzheimer's disease, which is typically something that occurs in older age, where there is degeneration of the acetylcholine neurons in the basal forebrain area as well as the brainstem. What you'll notice is that these areas that are indicated in these circles, and these very dense, are where the bodies or cell bodies of these neurons are, and then these arrows show you the projections in where they have their impact. You'll notice there's some in the brainstem, there's also some here in the forebrain area, and then they feed out into the cerebral cortex and have an overarching effect throughout the brain. Moving into the dopamine or dopaminergic pathway, this one has 2 primary pathways. One is the nigrostriatal, which has to do with movement, and when we talk about movement, we'll spend more time there. But think about Parkinson's disease. That is one neurodegenerative disease that we're aware of when the neurons in the basal ganglia and the substantia nigra start to fail or degenerate, that this is where we start to see the symptoms for dopamine. The other primary one is the mesolimbic pathway, and this is one that creates a major impact on our pleasure reward pathway. Any drug that is highly addictive will activate this pathway. So the 2 here, so you have this orange arrows here, this is the substantia nigra. This is the substantia nigra, I apologize. And then this is the pathway up to the caudate nucleus, which helps create smooth movements and helps our motor system function. This purple pathway here is the mesolimbic or the reward pathway is another way to think about it. It has an impact. It starts off in the ventral tegmental area, which is part of the midbrain, and then it feeds off into the nucleus accumbens, where it gets a major boost again. This pathway is connected to addiction, and any pleasurable feeling, whether or not you actually get addicted to it, will be activated here, and then it also plays a role in schizophrenia. The noradrenergic system, this is responsible for really making sure our body is activated and processing and able to respond to stimuli in our environment. It is one of the key players in the stress response system and managing whether or not we engage or retreat or withdraw from a situation. So it plays a major impact on our emotions. Does play a role or seems to have a connection between 2 extreme behaviors that we see in individuals, which is depression and mania. Depression is associated with a lower activity of the noradrenergic system, where mania is the opposite, where there's an excessive activation or high increased activation. The major source of this is the locus ceruleus, which is located in the brainstem, and then it has information that feeds off in both directions, where it goes down into the spinal cord and also plays a role in the cerebellum, where we have motor control, so it does play a role in motor movement, as well as feeding all throughout the brain and the prefrontal cortex. The serotonergic system, as I mentioned, has been linked with depression, and that seems to be the primary thing that most people go to when they think about serotonin, but it's actually responsible for a lot of other things in the brain and in our behavior. It plays a major role in our wakefulness and our ability to learn and learn long-term memory. It also has a major impact on our emotions, especially anger. So imbalances have been associated with depression, as we talked about, but also schizophrenia, OCD, and sleep apnea. So it has a major, it's just not a depression. It's way more complicated than that. The major sources of serotonin, or the reservoir in our brains, comes from the RAF nuclei, and then, as you can see, it floods and it has pathways that are linking to every area of our brain. Very important neurotransmitter. Before we move into learning and memory, where we're going to kind of apply some of these ideas to help bring home the message in learning these things, I want you to understand that those 4 neurotransmitters we just covered, cholinergic, dopaminergic, adrenergic, and serotonergic, those systems we talked about as being awakening systems or activating systems, they are responsible for helping the brain function normally and naturally, and keeping us awake and keeping us functioning, or inducing sleep. So that's why they mean by activating, whether you're awake and engaging with the world or whether you're going to sleep. It does not mean that they're all excitatory or they're all and they're not inhibitory. That depends on where the receptor is, what receptor you're binding to, and then the cascading effect on the neuron that it's connected to. So that's an important piece I want to drive home. The only 2 neurotransmitters that have very direct effects on the next neuron are glutamate and GABA. Glutamate's excitatory, GABA's inhibitory. Right, so let's look at just a few concepts that we're going to touch on for learning and memory. We're going to revisit these in section 3, but I feel like it helps bring things home. So learning is creating a long-term behavior change based on experience. And this we can see is done through neuroplasticity. Neuroplasticity is that concept that our nervous system has the ability to change and adapt. And this is the underlying, we theorize this is the underlying physiological changes that are connected to learning and memory. So habituation occurs when a stimulus is repeatedly provided and we desensitize to the effects of that stimulation. So again, habituation is a stimulus occurs and our response desensitizes or becomes weaker. How we see this happening occurs at the synaptic level, where you have the stimulus come in, the calcium influxes, and then you release a neurotransmitter, right? That's how a normal system functions. That's how a normal neurotransmitter synapse interacts. What we see with habituation is that the calcium goes in, but it does at a lower level, less influxes into the cell. Because the the calcium channels, their sensitivity to the change to positive 30 inside the cell is lower. Therefore, when you decrease the amount of calcium that's being brought into the presynaptic cell, you're going to reduce the amount of neurotransmitter that's produced. And that's on purpose because it realizes that sending that message repeatedly isn't helping or isn't making that process occur more quickly. And it can do it with less. Sensitization is the opposite. This is where the synapse has a greater response to the same stimuli. This can involve an interneuron producing additional neuromodulators or releasing additional neurotransmitters to have an impact. What has been seen, at least in animal models, is that the potassium channels open more slowly, meaning that the membrane, the neuron, doesn't repolarize as quickly, allowing that calcium to continue to influx into that presynaptic neuron, which increases the amount of neurotransmitter that's released. Habituation and sensitization are the exact opposite of each other. Sensitization is more calcium ends up getting into the presynaptic, which allows more neurotransmitter to be released. Habituation is less calcium being incorporated, therefore less neurotransmitter is being released. And all of this work was really done on the aplasia, which is a fantastic organism. We see is that in a situation where it becomes habituated, it actually removes the amount of stimuli that's ongoing. And when it's synthesized, there's actually an increase in the number of communication connections. Again, this picture is off because this communication is happening out here at the dendrites. I want to make sure I bring that home to you guys. Sometimes textbooks aren't perfect. All right. Last thing I want to kind of tap into with regards to learning, and we're going to talk about this more in depth, but I feel like it helps bring home some of these ideas. Neurotransmission relies on a lot of different factors that are being present, and when learning is occurring, what we find happens is that there's actually a second messenger. So we're using the metabotropic receptors, and that typically glutamate is one of those. It will create its secondary messenger. Its responsibility is to go off and actually interact with the DNA to create more receptors. So when learning is going on, you're actually increasing the number of receptors to facilitate communication with that neuron, with that presynaptic neuron, which is enhancing the connection between those two neurons. And that is what we believe is learning, is that you actually change the number of synapses and the number of receptors. It's an amazing kind of fluid process. And I want to introduce it here because we're going to come back to it when we get to learning and memory. And that's the end of this lecture.